All right, so now that we've got a good basis of RNA being the primary focus of transcription, let's start talking about the synthesis of RNA from DNA and really how this has led from an evolutionary perspective. So here we have an electron micrograph, and they look like tree branches with leaves coming out of them. So the tree branch is going to be the DNA, or the trunk of the tree is going to be the DNA, and all these little limbs coming off of them is actually RNA being transcribed from DNA. So this is what it looks like under an electron micrograph or electron microscope. So RNA is not going to be synthesized in one big mass like DNA is when we think of DNA replication. When we think of transcription and the production of RNA, that's only going to happen at the gene level. So certain genes are going to be turned on and transferred into messenger RNA at certain times. So, for example, here in this karyotype, if we've got this portion of chromosome 2 that codes for a gene for a specific trait, and the DNA code is this code down here in green, then we would be creating an RNA code as shown here in yellow. Basically, it's going to be the complement to the DNA, but instead of thymine, you're going to have uracil replacing it. So this production of messenger RNA is really a highly selective process. And the whole reason that we don't produce mass amounts of mRNA for entire chromosomes or entire genomes is because it's really inefficient. You don't need every single gene to produce a protein at every single point in time. So transcription is really tissue and gene specific. So there's three major requirements, of when we're synthesizing RNA from DNA. First of all, you need a DNA template to serve as the roadmap. So that's going to be the transcribed strand, and within that DNA template resides a transcription unit that has several subcategories, which we'll talk about in just a minute. You also need substrates or raw materials to actually build the new RNA molecules. So this is going to be similar to DNA replication like we talked about in chapter 12, just not as many substrates are going to be utilized. And finally, you need a transcription apparatus. So in bacterial cells, we have bacterial RNA polymerases, and in eukaryotic cells, we have eukaryotic RNA polymerases. So as we look at the template DNA, this template is going to be a single strand of DNA. Therefore, when a gene is being turned on, the first step is to form the replication fork and the replication bubble, and to get a single strand of DNA that isn't connected to its complement. So RNA synthesis happens in a complementary and anti-parallel form to the template strand. And the new nucleotides are always going to be added to the 3' OH group, just like in DNA replication. So the transcription proceeds in a 5' to 3' direction, very similar to DNA replication. And finally, the non-template strand is usually not going to be transcribed. So this is where we see our first major deviation from DNA replication, is because in transcription, the non-template strand nothing's going to be happening to it for the most part. Usually we don't use it. It's just the template strand is going to be coding for the gene. So the template strand is going to be a single strand of DNA, and transcription is going to use one nucleotide uses one nucleotide strand of DNA. And during transcription, RNA is going to happen again in a complementary but anti-parallel form, similar to DNA replication. So the DNA template can actually be housing several genes within a short amount of space. So RNA is going to be transcribed from one DNA strand. So different genes are going to be transcribed from different strands and even different locations on the chromosome. So for example, in this figure here, we've got, we'll just say this top strand is like chromosome 1, and this bottom strand is the complement to chromosome 1, and that first portion in orange is going to code for gene A, yellow is going to code for gene B, and purple or fuchsia, whatever color that is going to code for gene C. So gene A is going to go through the transcription process by moving from the 3' to the 5' direction. And then, but gene B is going to be moving from the 3' to 5' direction, but using the other strand as a template. So with DNA, with the double helix DNA, we tend to call one strand a negative strand and one strand a positive strand, just to keep the orientation correct. So our transcription unit is our first major requirement of transcription. So the transcription unit is going to be a stretch of DNA that codes for an RNA molecule and includes three major areas. It includes a promoter area, as shown here in yellow. It includes the DNA coding sequence, as shown here in purple and red, very important. So both of these segments together are considered the RNA coding region. And finally, the terminator is also going to be within the coding region, but is the third major part of that transcription unit. So the way that we orient this transcription unit is by first starting with our DNA strands. Our first strand is going to be oriented 5' to 3', and our second strand is going to be oriented 3' to 5'. So our top strand is going to be our non-template strand, whereas our bottom strand is going to be our roadmap for the transcription process. So the promoter region lies upstream of the start site. So here's our start site right here with the green arrow. And our promoter region is shown here in yellow, is upstream from that start site. Then the RNA coding region and the terminator region is going to be downstream from the start site. So after the transcription process has taken place, then we're going to have this messenger RNA strand that is going to contain the RNA coding region, which includes the termination sequence. And it's going to be oriented 5' to 3', the opposite of the template strand, which is oriented 3' to 5'. All right, so the transcription unit is, again, separated into those three major areas. So let's first talk about the promoter region. Again, that promoter is going to be oriented upstream from the start site, and this is going to be the area that is recognized by RNA polymerase. It's also going to show which strand is going to be used as the template strand, and it provides a direction in which RNA polymerase is going to move to synthesize the messenger RNA. And at the end of the promoter region, it's going to end with the start site. Sorry, let's go back. With the start site here. And that is going to show RNA polymerase where to begin synthesizing the messenger RNA strand. The RNA coding sequence, as shown in purple here, is going to be the sequence of DNA nucleotides that is copied into messenger RNA. And finally, the terminator, as shown in orange or red down here, is going to be where transcription ends. So that's like the end of the gene. So that's going to signal to RNA polymerase, hey buddy, you're going to transcribe this short segment and then you're done. So the transcription unit is going to be oriented in a 5' to 3' direction, but keep in mind we're always going to be adding nucleotides to the 3' OH group. So as we look down here at the bottom of this slide, If we have a DNA sequence that's written out in a non-template sequence, 5' to 3, our non-template DNA, like the top one up here, may have this code associated with it in the 5' to 3' direction. The template DNA, which you can equate to the second strand in our figure here, is going to be the complement to that non-template DNA. Therefore, the RNA sequence is going to be similar to the non-template sequence, except U is going to replace T. All right, so that's the easy way to remember this. The messenger RNA sequence is going to be very, very similar to the non-template DNA sequence. The only difference is that T is going to be replaced by U. So when we talk about upstream versus downstream, this all has to do with the location of the transcript start site. The first nucleotide is going to be numbered +1, and that's going to be downstream of the start site. Okay, and then anything that is downstream of that start site is going to be signified by a plus. Okay, so we have +1, which is going to be the nucleotide right after the start site, and then we may have +35 or +300 downstream. That's going to indicate the number of base pairs after the start site. The opposite is true before the start site, and those nucleotides are going to be designated as a minus or a negative. So one thing that we're going to look at in the next segment is how this promoter region is laid out. So we have certain sequences at -10 and -25 before the start site that is really important for RNA polymerase to recognize. So make sure that you have a really good handle on what is considered upstream and what is considered downstream of the transcription start site.